Okay, I'm going to take these headphones off because I look silly enough the way it is. I don't need headphones on. I'm coming to you from home, just like you, hopefully staying home and doing what we can to keep this virus from spreading. We talk a lot about viruses and spreading, and it's here. And you hear a lot of things that we talked about on the news that we talked about in the classroom. And it's good to at least be talking to you in one way or another and you to see and hear from me. I miss all of you. I wish we were back to normal and had our normal problems of being loud and tapping on desks and chewing gum in classroom. I would take all that over this right now. All those things are, seem very less important now that we have this going on. So I wish I could be there back in school with you, and hopefully we will at some point soon. Looks like at least a month here, I would say, with some new guidelines coming out. So, if any of you need anything, please reach out to me with an email or a comment in the Google Classroom. If you have technological concerns or any other concerns at all, just questions in general, I will honestly answer them the best I can. I am You know that I'm always honest with you. I'll give you any answer that I can, whether when it comes to school or this class or anything in general. I'll do my best to answer you and help you out as much as I can. One of the things I think that would be helpful to you is to try to get back to some sort of normal routine, and that is some class time. So what we are going to do is, after a review of mitosis, we are going to move forward in our curriculum because the meiosis stuff that we planned on doing, we don't have to do in 7th grade. So the meiosis stuff really is enrichment. The only thing we can be doing right now is review, remediation, and enrichment. And meiosis falls under enrichment because we don't really have to do that in 7th grade. We have to cover mitosis, but not meiosis. So we can go forward with what we had planned, which is nice. A little bit of normalcy here. Before we do that, we do have to review mitosis today so that you understand it, because meiosis is pretty much mitosis twice, which gets a little confusing. So if you don't understand mitosis, if you forget it, you're going to be lost when we start meiosis. So today we're going to go over mitosis. I'm going to try to stop making that mistake. Mitosis as a review. Make sure you understand this for tomorrow. If you need to go back and look at the videos that are in the classroom, which instead of pointing to them in the air, I can just say that they are, if you remember, they are right here. There's 2 of them right over there somewhere. But today we're going to go over mitosis as a review. I just have to find it again. That's not it. There we are. Okay. All right, remember there are several steps. It's interphase, PMAT, and then cytokinesis is sort of a little sub-process that happens along the way. Your cell spends most of its time in interphase. That's where it's growing and developing and getting ready to do PMAT, which is only about 10% of the time. What's happening in interphase? The DNA begins to duplicate. There are 46 chromatid. Yes, 46. Don't want to mess up the numbers. 46 chromatid, and they begin to duplicate. The cytoplasm begins to duplicate, and the organelles duplicate. Everything begins to duplicate because remember, we're starting with 1 cell, and we're going to pinch down. We're going to put 2. 2 body cells for your skin, your hair, your muscles, your nerves, your tendons, your ligaments, anything that has to deal with growing, repairing, developing, anything like that. These are not reproductive cells. That's going to be meiosis next, but these are body cells. So once everything is duplicated, interphase is over. Now remember, in the drawings, all the organelles are still here. We're just not redrawing all those organelles. What happens as we have after interphase is PMAT, and that stands for prophase, metaphase, anaphase, and telophase. In prophase, the nuclear membrane begins to break down, and all of the chromatid have created an identical duplicate of themselves, and we call that a sister chromatid. Now, that sister chromatid brings our total of chromatid up. We started with 46. Everybody makes a duplicate, and that means we have 92 chromatid now. Remember, we count the center. The center where they're attached is called a centromere, and we still count them. So technically, I have 46 chromosomes there, but I have 92 chromatid, okay, because everyone made a sister chromatid. The centrioles begin to form at the poles, and the spindle fibers begin to form. Remember, that's sort of like the fishing pole, and the spindle fibers are like the fishing line, and the centriole will be like the pole and the reel. Okay. Once we get all the chromosomes lined up in the middle, that is now considered metaphase. Metaphase is when they're lined up in the middle, and the spindle fibers are beginning to attach to the centrioles. What's going to happen then is that's all attached, so we try to get an equal number of chromosomes going to this pole and to that pole. If the organelles or the cytoplasm doesn't exactly duplicate and separate perfectly, it's okay. If we end up with 8 mitochondria on one side and 7 on the other, it's not a big deal. We have to have 46 chromatid on one side and 46 on the other. As soon as those chromatids start to move away from each other, that is anaphase. Okay? Remember, M for middle, A for anaphase. Once they start to move away, anaphase begins. Also, during anaphase, cytokinesis also begins. Cytokinesis is that pinching in of the cell membrane. Right? When you see a cleavage furrow, that's what we call that little dip, that little pinch in the cell membrane, you know that anaphase has begun and cytokinesis has begun. The spindle fibers begin to retract, and if all went well, 46 chromatid are going towards this centriole and 46 chromatid are going that way. Okay? All right, moving on. In telophase, the cytokinesis continues. That cleavage furrow gets deeper in, and the 46 chromosomes have made it here, and 46 have made it there. Now, to keep them from getting away and oozing around the cytoplasm, the nuclear membrane reforms. Okay, it keeps them there. Also, what begins to reform, you'll see the nucleolus. And remember, the nucleolus reforms because they make ribosomes, and ribosomes make proteins. And if we're going to start making 2 new cells, we're going to need proteins for building. So it's important that nucleolus gets back up and cranking out ribosomes so proteins can start being made. After telophase is full-on cytokinesis, which is right over here. That's when you end up with 2 daughter cells. Remember, that's the name of it. You have the original, and then you have a daughter cell right here. They are genetically identical because those chromatids duplicated, so they are identically the same. Pretty much interphase starts all over again at this point. As soon as cytokinesis happens, there's a brief rest period, but then interphase pretty much starts. It's going to start to duplicate the DNA again, duplicate the cytoplasm, duplicate the organelles. And if you recall, there was a slight difference in our plant friends. They have that rigid cell wall, so they cannot do cytokinesis by pinching in. Well, remember what happens is that plate begins to form in anaphase, and it continues to grow across and grow across and grow across, and then that will split into 2 plant cells. Okay? All right, well, that's a pretty quick mitosis review. I would suggest also going back to our website. If you remember how to get to that, it's click on the waterfall. And there are 2 helpful videos right there from the Amoeba Sisters and another one on Crash Course on Mitosis. Go back to your own website if you created a mitosis page and take a look at that. And tomorrow we will begin meiosis. Okay, that's it for me for today. Hopefully the rest of your classes go well. If you have any questions at all about our class, your other classes, or anything, reach out to me via email, the Remind app, or in the Google Classroom, and I will get back to you. I will be sitting in front of my computer 9:30 to 10:30, checking for emails, and then also 2:30 to 3:30. Those are what we're calling office hours, where we are there. If you email me between, I'll definitely be checking my email and other spots like Remind and Google Classrooms in between. But for those 2 solid hours, that's all I'll be doing. All right. Well, I hope everyone is well, and I'll see you in the next video. Bye.